Thank you, Peter, and welcome everyone. So as Peter mentioned, I'm joined here today by Sanjita Rangwala, and Valerie Schneider's on the phone. She's the lead of the group that works on the sequence viewers at NCBI. And we also have Ravinder with us in the room today to help answer questions. So the idea today is in part to advertise the fact that we now have Track Hubs available in NCBI's genome browser, the Genome Data Viewer, and give some demonstration of how to find Track Hubs and then how to connect to them and see what they look like in the displays. So today's topics are going to be what is a Track Hub? Maybe some of you are not familiar with Track Hubs, so I'll give a very brief introduction to what a Track Hub is. And then still in the slides, show you how you can search for Track Hubs within the Genome Data Viewer and a bit on how to connect to those and display those. And then I'll jump into the viewer itself and do a live demo, just emphasizing some of those features. And I'll wrap up with some general features of the Genome Data Viewer, such as how to share your browser view once you've configured that. So let's start with what is a Track Hub. And I put this reference up here for two reasons. One, it's a great source of background information on what Track Hubs are. So if you're not that familiar with them, this would be a good place to get information. It also reminds me that Track Hubs are not an NCBI product, so don't want there to be any confusion about that. These were developed by the good folks at UCSC. So from a user's point of view, a Track Hub is simply a method of displaying annotation data.
These data can be epigenomics, proteomics, they can be expression data, features like transcription start sites, that sort of thing. These are typically pretty large files in formats like BigBed, BigWig, and BAM. And the real advantages of Track Hubs are that the data are on a remote server relative to you so that you don't have to maintain the data. And also, whenever you ask the browser to retrieve a certain bit of data, it only has to retrieve what is being displayed in your browser at the moment, that range of genomic coordinates.
But also, whenever you ask the browser to retrieve a certain bit of data, it only has to retrieve what is being displayed in your browser at the moment. In other words, that range of genomic coordinates. So it doesn't have to pull everything that's in that project. It only pulls what it needs to show in that particular view. And so that makes loading the tracks pretty fast. So as you can imagine, as more and more Track Hubs have become available, there needed to be a way to organize them and have a good search interface to find them. So the Track Hub Registry came into being, primarily from the folks at EMVL and EBI. And I encourage you to look at this website, trackhubregistry.org, to get more background on that. And a good point of our Genome Data Viewer is that we provide a search interface so you can easily search just using free text this Track Hub Registry. So I'm going to start here on the Genome Data Viewer homepage, and I've blown up the section here on the right where you can choose the assembly. And that's to point out that you do need to make sure you're working in the correct assembly version for the annotation data that you want to use. Annotation data is, of course, mapped to a particular version. So the Track Hub Registry has many different organisms, and it often has more than one assembly for a given organism. So make sure you choose the correct assembly there. So once you do that, you can either click on that Browse Genome button, or you could use the search box up above to locate yourself to a particular chromosome or region on a chromosome or a gene or something like that. Let's just say you click on that Browse Genome button. You're going to get a view like this of the Genome Data Viewer. And on the left-hand side of that page are what we call widgets. So there's a search widget and a blast widget. And the one we're going to be talking about today and using is the user data and Track Hubs widget. So to expand that a bit, within that widget is an options menu. You can see expanded on the right there. And under options is a selection for Track Hubs. This is also, by the way, where you could load up your local files of user data. So if you click on Track Hubs, that's going to take you into a Track Hub configuration menu where you can start your search. And so here you can input the URL for the hub if you happen to know that. You can also restrict by data type if you want, genomics, transcriptomics, proteomics. In many cases, just searching any of those is fine. And so let's say I knew that I wanted hubs associated with the GenCode project. I can simply type GenCode in there, hit go, and on this particular day, that retrieved 3 different hubs. And keep in mind, this is from the GRC H37 or HG19 assembly. And so you have GenCode related data of different types, long non-coding RNA race data, predicted microRNA target sites, and a project called CSF. So let's say I'm interested in that. I check that box. That changes the display of the menu and then lists all of the tracks that are in that particular hub. And I'll show this in more detail when I get into the live demo. But at the bottom of that list, it also shows you other assemblies on which this Track Hub is available. So it's a little difficult to tell on the slide, but those are grayed out because they wouldn't be able to display in the HG19 assembly. But at least you know that these are available. So there's the other human assembly, HG38, the current assembly, and then assemblies for mouse, chicken, fruit fly, and C. elegans. And then when you select the tracks that you want to load up, you just check a box for the tracks that you want. You get a nice green light on the right there when the tracks load up, and then you hit the configure button, and it adds those tracks into the bottom of your view. Those should be displaying as green tinted tracks. They're not on my monitor for some reason, but it loads 2 tracks at the bottom, the 2 that I had requested. And I have zoomed in here to a particular gene. And if we wanted to look at that in more detail, we could zoom in even farther. And so we see here that the splice donor predictions are lining up with one end of an exon and the splice acceptor predictions with the other. So that looks good. So that's all I had to do for the slides. I'm going to now jump into the Genome Data Viewer and do a bit of live demo. So here I am on a browser in the Genome Data Viewer. I just want to back up a bit and give you a little perspective on why we think using track hubs is important. So we have this. Let me increase the zoom level on this page a bit. So you'll see up here at the top, we have this nice array of over 600 eukaryotic RefSeq genome assemblies in their annotation. And so as more people put data into track hubs, you'll be able to align this other source of data in the same viewer with the NCBI annotations. With the overall goal is that having 2 or more different sources of annotation might be able to come up with some new insights or at least make some better annotations. So that's sort of the overall goal here. With that little introduction, I'll jump into the Genome Data Viewer and see how these track hubs can help facilitate that goal. So I'm going to go ahead and choose GRC H37. There are, in fact, many more hubs available for GRC H38, but I'm just using this as an example. I'm going to click on Browse Genome, and that's loading the page. Okay, so as always, the viewer comes up with a default set of tracks. Let me see if I can zoom in a bit more. So I'm going to close a couple of these widgets over here right now and open up this user data and track hubs widget. So I've got no track hubs or other tracks right now. So if I want to go to that configuration menu, I go to the options menu and click on track hubs. And so now I can search for any number of things. And this is a very versatile search interface. So let's say I want to pull up that Philo CSF project, and maybe I don't remember the whole name there. I can use wildcards here, so Philo asterisk, and hit go, and it finds that project for me. So that's a nice feature. Now I check that box, and again you're going to see the list of tracks here that are available for this particular hub. So we have big bed tracks, big wig tracks in this particular set. And you may notice that some of these tracks are in red. So when a provider puts together his track hub, submits it to the Track Hub Registry, he can mark a certain set of tracks as a default set of tracks. So all of these up here in black are the default set. These others are not marked as default, but you can indeed load these up if you want. So, maybe these are sort of some type of supporting data. For example, this Philo's CSF power is a measure of that metric statistical power. That's what that red color is about. And here are those other assemblies that I mentioned. Okay, so let's go ahead and pick the Philo CSF novel set. So I'll check that box, and you get some configuration options here, but you do notice that you get this green dot. Those of you paying attention will see that Track validated with issues will eventually, of course, fix this, but this is an issue only with chromosome M, the mitochondrial genome. So it's not going to affect the ability to load up a track for the nuclear genome. So that looks good. And I will go ahead and click Configure then. And before we look at the sequence viewer, I want to point out over here on the left, so it now displays that track that I loaded, and there's this very handy description link here. So if you want to learn more about this particular project, you can click on description, and this is provided by the hub provider. And so each of the different types of tracks are discussed here. And so we see that this novel track, for instance, shows potential protein coding regions that are not currently annotated as protein coding exons. So that's what this particular track is all about. Okay, so I think what I'll do is clean up some of the tracks here. We see our Philo CSF novel regions down here at the bottom. And by default, the viewer comes in, at least for human, showing all of human chromosome 1. So this is all of chromosome 1. But let me get rid of some of these other tracks here just to clean this up a bit. Yeah, I can get rid of that one too. And you may or may not know this, you can also move tracks around. So if I wanted to put this hub track right up there below the genes track, I can do that. But let's say I want to focus in on a particular gene region. So I can go back up to the search widget, and let's look for the CSK gene, the C-terminal Sark kinase gene. So it's now going to focus in on that region. Okay, and so here we have our track hub. I moved it up so it's in between the NCBI gene annotation and the ensemble gene annotation. And so these are the features that come with this particular track. You can mouse over those, you get some more information. If you wanted the exact sequence of that region, you could get that out of here with this FASTA view. And these are colored differently based on the strand that they're on. So I think green is plus strand, red is minus strand. So we see some features in this gene that are showing up in regions that some are and some are not. In regions that are currently annotated as protein coding. This is actually a 5' UTR region up here. And of course, you can zoom around in the browser if you want. You could zoom out and see what this looks like on nearby genes. Okay, so let me check my notes here real quick to see if one. Let's do another search and we'll add another track. So I could go back either through this Track Hub's menu, or in fact, I could just click on this configure button here, and that will get me back into this search interface. I could, close this up if I want to clean it up. But to do another search, all you have to do is click on this magnifying glass here. And let's say we want to add a track for epigenomics. Type that in there. Hit go. And so in GRCH 37, that shows us 4 tracks. I'm going to ultimately pick this top one here, the Cottony Lab, but I want to show you another feature here. So if I want to search, let's say I'm specifically interested in looking for DNase signatures. So I can just search for DNase. In this particular case, in this assembly, it comes up with not exactly the same sets, but it does include this one. And so I'll check this one for the Cottony Lab, and we'll connect to that one. And so now we see all the tracks available for that. So many of these are not part of the default set, but there's a large number of possibilities to choose from here. You can, of course, narrow things down here by closing these carets. And I'm not sure what other genomes this is available on, but we can go down to the bottom. So it's also available for mouse. So let me go back up to the top if I can get my mouse to cooperate. So let's just put up this impute CS17 combined DNase track. This is a big WIG track now, not a big bed track. So I checked that. I've got the green light. I'll click configure. And there now it's added in this track. So if I want to go back and look just at that CSK gene, I can use the region menu up here and say go to gene with pad. Pad is just a little space on the side. And so now, let me move this track up now. So it's right under our annotation there. Okay, well, it's right under the ensemble annotation. But we have a nice signature here, right at the beginning of this 5' UTR. And these show up pretty nicely in the surrounding genes as well. So that's the basic idea. You can add multiple tracks that way, and you still have the options of configuring in a couple different spots. So let's do one more thing here. I will, let's say we want to look at this phylo-CSF in Drosophila for the same gene. So I'll just right-click up here on the title and open a new browser window for the GDV homepage. And I got to make that a little smaller. And so now I want to search in fruit fly. And there's only one assembly in fruit fly, so I don't have to worry about that. So I'll just go ahead and click on Browse Genome. And let's go ahead and narrow that to the same gene region in the search box here. That's loading up. Okay, so we've got the CSK gene there. Let me go ahead and clean this up a bit. All right, so when I open up the track widget here, and let me zoom in a bit. So I should already have that phylo-CSF track available because I called it up earlier in human. So I can select that. And I could get a description if I wanted to, or I can just go ahead and say configure. That will take me into the menu. And so now I'm looking at the Drosophila melanogaster 6 genome annotation. So I will check that box. Got the green light, so I'll click on configure. And there we have very similar sort of annotations there. It's in a little different spot. It's amazing the CSK gene is this well conserved between human and fly. If I wanted to, I could move that up again. Okay, so that's it for that demonstration. I really just want to show you a couple more things, general features in the browser. And so let's say you get this configured exactly the way you want, and you want to show this to one of your colleagues. There's a link up here, and let me zoom in on this a little bit more. Link up here at the top of the browser called Share This Page. So you click on that, and it will generate a URL. So you send that URL to one of your colleagues, and they will see the exact same view that you're seeing here. Other thing you can do, of course, is use the Tools menu. Scroll over here a bit. And so here you can print this as a publication quality PDF. You could send that to somebody. And I don't want you to get confused on the Share This Page link. You may know that within the sequence viewer, there is a link to view. You could use that, and it will still display these same tracks, but it will just be the sequence viewer. It won't be the whole Genome Data Viewer view, so you won't have the widgets over there. So I'm finished with my bit, and we'll open it up, see if there are any questions. Any general questions, Peter?
Thanks, Wayne. No, there are no questions right now. Please please be sure to, if you think of anything afterwards, please be sure to write to us. And Wayne, I think, has a slide that shows you some contact information here in a minute. So if you have any questions about the Genome Data Viewer or any questions about the webinar program in general, you can write to us. Also, some useful links for good resources for learning about NCBI, in particular the NCBI Insights. And you can sign up for announcements, and you can check out our YouTube channel where this video will live. So thanks for coming, and we'll talk to you next time.